Hello, everyone. Welcome to Lightning Talks. This is season 6, episode 3. We are coming to you live from the Seattle Aquarium on the shores of Puget Sound in the traditional and contemporary territories of the Coast Salish people. And we know we have speakers and viewers joining us from near and far. So thank you for joining us wherever you may be. My name is Nora Nickham. I use she, her pronouns, and I'm the Vice President of Conservation Programs and Partnerships here at the Seattle Aquarium. And tonight our episode is all about coral. And I wanted to introduce you to my personal favorite coral, which is called the spiral wire coral. This kind of coral is an animal like all other corals. It likes strong currents and it eats plankton and other bits of yummy things that it finds and can reach using the tentacles on its polyps. It also is a favorite of mine because it reminds me of the pipe cleaners in my daughter's craft kits. And we have several of these here at the Seattle Aquarium in our Ocean Pavilion. So I hope next time you're visiting, you can stop by and see them. And as we learned in the trivia tonight, there are many kinds of coral. So if you have a personal favorite, I hope you will tell us about it in the chat. Now, let me tell you how Lightning Talks works if you are new to it. We have 5 speakers. They each have exactly 5 minutes to share one big idea or topic. And they're going to get warning lights to make sure they stay on time. So they'll get this when they have 1 minute remaining. They'll get a yellow light when they have 30 seconds remaining. And they get a red light when they're at time. And then if they keep going, then so that's the lightning strikes that give Lightning Talks its name. Now, all along the way, I'm sure you will have lots of burning questions and thoughts to share. So please put those in the chat, or you can use the QR code. And we're going to bring all of our speakers back at the end for a great conversation where we're going to draw from the questions that you have submitted live. If you enjoyed tonight's talk, we hope you will consider a donation to the Seattle Aquarium. You would be joining us in our mission of inspiring conservation of the marine environment through lots of different programs, including Lightning Talks. So now I would love to go ahead and introduce our first speaker, welcoming to the Lightning Talk stage, Dr. Nadege Aoki. Nadege recently completed her PhD in biological oceanography at MIT and the Woods Hole Oceanographic Institution. Her thesis looked at coral reef soundscapes and the ecological role of sound in coral larval settlement. Nadege, are you ready to kick us off?
I am so ready.
All right. Your slides look great.
Okay, great. I'm going to get started. Thanks everyone for joining. I'm so excited to be having this discussion about corals. They're my favorite marine creatures. So today I'm going to be sort of talking a little bit about how corals are born, how baby corals find their way around the reef, and how sound and the sound of the reef is an approach that scientists are currently studying to try to help baby corals in that journey. So like Nora mentioned, corals are animals, and so many corals reproduce via sexual reproduction, where individual coral colonies release eggs and sperm into the water column, and then those gametes join together to create fertilized coral embryos. So this video shows a zoomed-in view of a Hawaiian cauliflower coral during a spawning event, and you can actually see that these little dots being released from the polyps, those are actually the coral eggs. And so different species of coral spawn at different times of the year, and those spawning events are usually linked to the lunar cycle. And so after those gametes fertilize, they develop into embryos. And after a few days of development, those embryos turn into coral larvae. And what's really cool about these larvae and makes them different from the adult form of corals is that these larvae are mobile. They can swim around the reef. They can check out different parts, different substrates, and decide what's the best spot to attach to and develop into an adult. And so this is the only time during a coral's life cycle when it has this mobility and ability to choose a different habitat. And that process of picking where to attach and develop, we call coral settlement, because the larvae are settling onto the reef. So obviously, it's pretty important that these larvae pick a good spot to settle, so somewhere where they'll have a really good chance of survival. And so to do this, larvae are using a whole bunch of different sensory mechanisms to gather information about their environment. They're smelling and tasting chemicals on the reef, they're sensing the light environment, and we've recently discovered that they're also listening to the sounds of the reef. So what does that mean? What does a coral reef sound like? And what could these larvae be listening for? Because reefs are such biodiverse environments, they're also really acoustically active because many of the animals that live on reefs, like snapping shrimp and many different species of fishes, actually produce sounds. So I'm gonna play a quick 5-second clip, and you can hopefully listen at home and hear in the background, there'll be these sort of crackling sounds that are from shrimp, and then these low pulses that are sounds made by fishes. Right? So it's pretty cool. And we know that these biological sound cues are more present and louder on healthy reefs compared to degraded reefs. I'll play it again. So as we're learning how the sound environment is really important for early stages of corals and other reef animals, scientists have wanted to test this question of, can we enrich the acoustic environment of a degraded coral reef to attract these juvenile animals and potentially help reseed and rebuild the reef? So can we put a speaker down, like in the schematic, and play the sound of a healthy reef and try to help rebuild that degraded site? So in my PhD work, I did a series of acoustic enrichment experiments on this topic. So I collected larvae from 4 different coral species and exposed these larvae to different sound treatments and then looked at how that affected their settlement rates. And so 2 of these studies have recently been published and you can go find them online. 2 more will hopefully be published in the next year. I studied corals from the Caribbean and the US Virgin Islands, as well as the Pacific in Hawaii. And so this slide just shows some sample results and some images from the setup. So on the left side, you can see these little kind of cups, like almost like a urine specimen cup that we put some larvae in to attach them to the reef. And then on the bottom image, it's our blue underwater speaker that we use to play the sounds to the corals. And this graph shows that at the red site, that was our playback site, the average settlement rate was about 2 times higher than at our control sites where we didn't play extra sound. So if you want to learn more about this work and other solution-based coral reef science, I highly encourage you to check out the Reef Solutions group at Woods Hole Oceanographic Institution. Thank you all so much. That's pretty much all I have.
Nice. No lightning strike for you, Nadej. That was so interesting. I'm just thinking about what a big decision it is for those coral larvae to decide where they're going to live their whole lives when they're little.
Yeah, and they're pretty picky about it. You know, you can, if you have them in the lab and you don't give them the right settlement cues, they will stay swimming and delay metamorphosis for a long time.
That's amazing. So I don't believe corals have ears. So how do you think they detect the sound?
Yeah, so this is something that we don't fully understand yet, but there are a lot of other invertebrates that don't have ears that are sensitive to sound, particularly the component of sound that's really vibrations, what we call particle motion of an acoustic wave moving through the water column. So we think that because corals have ciliary hairs, so they have these like sensory structures on their exteriors, that might be helping them detect the vibrations of the water. But we don't fully know, and we need more research to find out.
So interesting. Well, I hope all of our viewers with more questions for you will go ahead and put those in the chat or use that QR code, and we'll bring you back at the end for more conversation. And for now, we're going to bring up our next speaker on the Lightning Talk stage, Amy Olsen. Amy is a research scientist with us here at the Seattle Aquarium, and she studies everything from coral reefs and sea otters to water quality and microplastics. So Amy, we would love to hear from you.
Coral reefs are incredibly important to our marine ecosystem. They provide crucial habitat to many species, economic benefits such as fisheries and tourism, and have cultural value to many coastal communities. But they are very sensitive. Here are screenshots from one of our research sites in 2009 versus 2025, and this is the exact same point. And you can see that stressors such as warming ocean temperatures, increased wave action, and poor water quality have hurt these reefs in Hawaii. By capturing videos of the same coral reefs year after year, we are able to measure changes over time. But sometimes this can be a lot of data. So where can we get some help? My name is Amy Olson. I'm a research scientist at the Seattle Aquarium, and I'm here to talk about how we can train computer models to help with coral conservation. So the Seattle Aquarium has been monitoring 8 sites on the west side of the Big Island of Hawaii since 2009. And you can see on the map in the top left-hand corner, we have those 8 points, and they're different colors because each of these areas is managed differently. This project was designed with partners in the bottom left-hand corner. Some of these organizations have been involved since the very beginning, and others more recently. And we use a really unique survey method. We have divers that go down to our sites. One person's carrying a video camera, so it's diver-operated video that we're capturing, and the other person has the transect tape. So we have 8 sites that we survey once a year, and we have 18 years of data, so that is about 144 videos. Now for each of these videos, we take 40 screenshots, and in each of those screenshots or pictures, we randomly overlay 10 points on the bottom of the photo, on the bottom of the ocean, to analyze, which means for each video, we have 400 points. Now, who can tell me what 144 times 400 is? That's 57,600 points that we need to analyze. Wow. So yay for job security for me, but this would take forever. So what can we use? What tools do we have to use to be able to get this work done a little bit quicker than if I went through each of these points by myself? CoralNet has joined the chat. CoralNet is a free open source online platform used by marine scientists to analyze benthic images of coral reefs and other aquatic environments. It uses deep neural networks and machine learning to automatically classify what's in the photos. So if you look at the photo on the right, we have an image from one of our surveys, and those purple crosses are 10 randomly scattered points on the bottom part of the photo. First, a human has to go through and say, for under point 1, it's a hard substrate. Under point 2, it's a dead species of coral. 3 is a live, this species. So this is a fish, this is sand, and you're training the machine learning. And after time, the computer gets better and better and better at guessing what it thinks is under each of these points. So in the beginning, it's a lot of human-powered work to analyze these points. Eventually, the machine learning takes over and it gives educated guesses, and then a human goes through to double check all of these points. So using this machine learning, what have we found about how corals have responded to a series of marine heat waves, which are essentially periods of warm ocean temperatures in the west coast of Hawaii Island? And one of the most common species that we see is Parietes lobata, which is this lobe coral. It's very common in along the coast, but also at a lot of our sites. And what we saw was a decline in healthy corals after the marine heat waves, with a slight increase in the most recent years, which may indicate that they're starting to come back a little bit. There might be a little bit of resilience at some of our sites. This other species, Parietes compressa, is a little bit different. You can see it's more finger-shaped. There's a lot more surface area. It's a little bit more sensitive to physical breakage. So at a lot of our sites, unfortunately, we're seeing decreases over time after these marine heat waves, with continues of declining. So monitoring coral reefs for long periods of time is very important, as we expect ocean temperatures to increase and marine heat waves to occur more often due to climate change, and computer models can help. Thank you.
Thank you, Amy. Yes, thank you for emphasizing why having 18 years of data is important and why we need many more years going forward. I was wondering if you could give an example of how findings from this kind of research could help inform decisions about conservation.
Yeah, so none of the work that we do at the aquarium happens in a vacuum. We have many partners that I showed on my third slide. And so we share our data with many folks on the island, but specifically the Division of Aquatic Resources, the Kona office, which manages these different areas. And so we meet with them, we share our data and our publications with them so that they're able to make better informed decisions for their local waters.
Fantastic. Great to connect science with policy and management. Thank
Our
you,
favorite.
Amy. Our favorite. Thank you. We'll welcome you back in a few minutes for the discussion. And I hope there are plenty of questions in the chat for Amy. And in the meantime, welcome Emma. So our next speaker is Emma Orzek. Emma is an aquarist here at the Seattle Aquarium, and she cares for fish and invertebrates throughout our tropical environments. Emma has expertise running coral propagation and coral reef systems here and at Georgia Aquarium, where she also managed the Florida Coral Reef Tract project. We look forward to learning from you, Emma. Get your slides up. They look great. Take it away.
Great. So we learned earlier about how corals sexually reproduce, but this is a little bit about how they asexually reproduce, known as coral propagation, which is very common in aquarium industries. So what is coral propagation? It is making fragments of corals to promote growth or spread the colonies. It is very similar to propagating a plant, if you've ever done that, where you trim off a leaf and able to repot it, and then you can grow that individual. So here I have two examples of two different corals that I have fragged. You can see this top one is an Acropora, and you can see that I've planted it onto this little tiny tile to hopefully grow into a larger colony like its parent. And then this bottom coral, you can see that it used to be quite a few small corals that have grown into each other and will become one healthy colony. And how do we do this? So there are a lot of tools, a lot of different ways. Every coral is different. So we do have coral cutters, very similar to bone cutters that we can use to trim off branches or just trim off small pieces. We have a griffin saw, which is very helpful for cutting through larger corals if they're very thick skeletons. It can be a little bit more accurate if you are working to have like a very large coral. Or you could use a hammer and chisel, which is pretty common in the field on rock work, trying to trim off corals that way. Once the coral is trimmed off, we do use reef glue, icy gel, different types of epoxies to glue that coral down onto a new substrate. It could be anything from a plug, like the little items you see on the left, or a tile, or directly onto a rock or the reef wall itself. So why do we do this? It can be for coral health to prevent bleaching. So if you look at the two upper corals, you can see there was a little bleach spot on one side. Another coral fell on top of it. And just to prevent further spread of bleaching, I trimmed off that infected piece to hopefully help the healthy coral grow back into a healthier section. And then sometimes it is to spread the colony. So the very bottom coral is a Cereatopora. You can see the larger parent colony on the left and the smaller new baby on the right. So that's just something that I trimmed off. Hopefully it'll grow to be the same size as its parents. And sometimes we do it for the vibes. Sometimes I have a really personal connection with the coral. I like the color, I like the name. It grows really well. Sometimes I just want more of a coral, so I'll trim off a piece of it and see if I can get it to grow somewhere else and spread a coral that I really like and I'm passionate about for everybody else to see. The most common question that I get asked is timing. So how fast a coral can grow. So first off, this is a picture of me in April 2022. My mentor at the time, Zaylee, took this picture of me. It's my very first coral shipment. You can see this comes in a really small box. I had a really teeny tiny bag with water in it, and you see this little coral. It was about 1/2 inch. It is a rainbow Montipora. And then fast forward to May 2024. This is what it looks like in 2024. So you can see that original tile, the really small round piece in the center. And then you can see I had glued it onto another ceramic tile. It outgrew that tile as well. And actually I did trim off half of it and I fragged it quite a few times trying to make more of it because, again, I thought it was a really cool coral and I was happy to share it with other facilities and give them frags to try and grow it as well. And then I did get an updated picture from 2026. So this is what it looks like now, which is really exciting. It's about 10 inches tall, has spread to be about 12 inches. So from 2022 to 2026, you can see that it has grown significantly, which is really exciting. It has grown from these orange little polyps into half an inch into this large, beautiful green colony. And that's just one example. So this Montipora did grow a lot, but I have had like Acropora and Stereotoporas that have taken a lot longer to grow. Each coral is very unique in its own way, and you have to figure out what works best for the coral. And that's why we propagate them, to try and see if we can find better places for them to grow. Thank you.
Thank you, Emma. I have to say, I love the lingo of fragging coral, which I assume means making fragments.
Yes.
I love it. What are some things you have to be careful of when you're fragging corals? Like you would train somebody who was learning how to do it.
You want to be really careful that you're not taking too much of a colony, especially if it's a smaller colony. And when you are fragging a coral, you really want to be sure that you are getting polyps in the frag. Some corals have very spread out polyps, so you don't want to take a piece of a coral that doesn't have a polyp on it because it would not be able to grow successfully.
Okay, good tips. Thank you so much, Emma. We will see questions coming in for you and we'll bring you back in a little bit. And for now, we are going to bring up our next speaker, Dr. Trinity Kahn. Trinity is a postdoc at the Shedd Aquarium in the Conservation Research Department, working with Dr. Ross Cunning. Trinity has a PhD in biology from the Pennsylvania State University, and she studies how the genomes of reef-building corals evolve over time and how they may respond to environmental change. So we'll get your slides up. Just need to get them full screen. There we go. They look great. Go ahead, Trinity. We're not hearing you, Trinity. Hold on just a second while we fix the audio. Can you try saying something? Yeah. Okay, yeah. Now we can hear you. Okay, let's get those slides back.
Yep.
All right, go for it.
Hi, sorry. So like she said, I'm Trinity. I'm a postdoc at Shedd Aquarium. And today I'm going to talk a little bit about our work using genomics and genetics to understand what drives thermal resilience in corals and how we can use that to make more resilient reefs in the future. So we sort of touched on it a little bit, but just as sort of overview, probably the largest threat to corals and coral reefs right now is climate change and rising ocean temperatures. So when beautiful coral reefs like this are healthy, you see a lot of color on the reef. And then if it gets too hot, something called coral bleaching, where they expel their algae and turn white happens. So this is basically the coral starving. And if the stress gets too long and they stay bleached, then eventually you'll see mass mortality on these reefs. So this is sort of one of our biggest concerns in coral reef conservation. And a lot of the basis of bleaching is because of this partnership that they have with the algae in their cells that helps give them food. So this partnership is both individuals driving whether or not a coral will bleach under certain temperatures or not. But today I'm going to focus on just the coral host. And specifically using the genome of the coral host to understand what drives why one coral may bleach and the other may not. So to do this, me and my colleagues at Shedd Aquarium embarked on a sort of pan-ocean research expedition covering Florida, American Samoa, and Moorea to look at 3 different species of corals to really try to understand, okay, what in the coral genome drives why some of them can handle higher temperatures and why some can't. So we did this. We looked at 3 different species of staghorn coral. They're a very sort of shallow, fast-growing coral that's really important for the ecology of these reefs. And they all sort of also play very similar roles, have very similar morphology. So using all three of these staghorn corals, we actually then measured their thermal tolerance using sort of a in the field party cooler based acute heat stress assay. So we heat them up really fast, and then we can actually measure how thermally tolerant they are. So after measuring and performing those experiments, so we have two individuals of corals. We can then say, okay, this one coral has a thermal tolerance of 34.5 degrees Celsius, and this one has one of 36.1. And then we see a large variability in this thermal tolerance. So we see, individuals all across this distribution of values. And now we want to know, okay, why? And to do that, we can sequence their genomes. So we look at all three of these colonies across the distribution and can actually sequence the entire genome of each of these corals and analyze all the different nucleotides that make up those genomes and identify which of those are associated with where on this distribution those corals fall. So I'm going to talk about something called a polygenic trait. So thermal tolerance, when we were sort of first looking at this, we really wanted to find the gene. I call it gene fix it all. This is the gene that drives whether a coral is going to bleach or not. Any change in that gene is what's going to determine whether these corals are going to survive or not. So in ideal world, we would turn the gene on or have one nucleotide in that gene to a higher thermal tolerance, and another version of that gene leads to a lower thermal tolerance. That is not what we find at all. We actually find it's a polygenic trait, which means lots of different thousands and thousands of different places in the genome actually dictate together what kind of thermal tolerance a coral will have. And so we can actually take that information, look at all of the different letters, all the different nucleotides across the genome, and use that information together to then predict thermal tolerance from that genetic data. So using only a tiny tissue sample, we can then predict how thermally tolerant an individual might be. And we find that when we predict that thermal tolerance, it's highly correlated with what we've measured, which means that, okay, we can actually go forward and use this in conservation and restoration applications. It's not just numbers. And so using this information, we can actually measure the thermal tolerance of two parents or predict it, and then actually predict how their offspring will perform. So then we can design selective breeding experiments to produce offspring in sexual restoration programs that will then be able to be of certain thermal tolerances, or we can predict what the reef will look like after certain restoration actions. And this is sort of getting us one step closer to hopefully building more resilient reefs with our work. Thank you.
Thank you, Trinity. So directly applicable to important conservation work. That's great. I was curious when you were showing the corals in the party coolers, how quickly can you tell if they are intolerant to the water temperature, and what does that look like?
Yeah, so it's a really good question. So the experiment we run is actually just over a 12-hour period. So it's a really fast, and we heat some corals up to like 9 degrees over their maximum. And you sort of start to see maybe 6 degrees. So it's like way more than they would ever actually experience. But you start to see certain individuals, staying colored, not bleaching at all, usually about at maybe 5 degrees above their average temperatures within a few hours. And so it's like a really great visual of how much variance there is in these genomes and these corals to different temperatures. So it's really quite quick. It's amazing.
That is really amazing. Well, thank you, Trinity. We have lots more questions for you, so we're looking forward to bringing you back in a few minutes and reminding our viewers to use that QR code or put your questions in the chat. And our final speaker for this evening is Dr. Greg Asner. Greg is the director of Arizona State University's Center for Global Discovery and Conservation Science. He's based in Hawaii, where he oversees ASU's science activities. Greg is an ecologist recognized for his exploratory and applied research on land and ocean ecosystems at regional to global scales. We're looking forward to your talk, Greg.
Great. You can hear me loud and clear?
Yep.
Great. Well, I figured by the time we get to me, we would have heard a lot about coral, and the 4 speakers covered all of the details. And so I'm going to zoom out, and I'm going to talk for 5 minutes about what we can do to put what you just heard together at a community level to do some interventions and to try to save these reefs. I'm going to take us back to West Hawaii, and I'm so glad that we heard about West Hawaii earlier. I'm based here in Kailua Kona. You see this map here. The western side of the island is down on this map. And these red dots and yellow dots and green dots are showing us the relative condition of this reef. And so what we've done in this largest contiguous reef in the state of Hawaii is develop what we call the Long Reef Medical Center. It's got a name called Akoakoa in Hawaiian that means for people to come together. And also it has a second meaning in the Hawaiian language for corals to assemble on the seafloor. So that's where we get our program's name. You already heard about our marine heat waves causing us coral bleaching. You already heard about wastewater. For 23 years, our team has been monitoring the entire west coast of the Big Island, wastewater, urban runoff, nutrient loading, all of the vital signs of this reef. And what we've come to figure out is that this combination of factors really is requiring us to think about holistic interventions that bring us back into the ecosystem as partners with the reef to try to develop and generate a new future for it. To do that, what does a holistic program look like? It has cultural leadership. It has the sciences that you've seen tonight. It has education for our visiting community and our residents, and strong government interactions, partnerships, new laws, new regulations, all sorts of things going on. Altogether, this has to happen. The cultural leadership is front and center in Hawaii, especially on the Big Island and some of our outer islands. It really is the lifeblood of why we're here, what we're doing here, and the story of our place. The science is a toolkit to the community in this kind of context. The cultural leadership is first. The science serves that leadership. The science is pretty advanced when you're dealing with a 200-kilometer long reef. This is our aircraft system that's in its 20th year. It's a highly advanced regional platform that allows us to measure the reef in unprecedented detail all the way to the bottom of the edge of the reef where the corals sort of end and the deep ocean begins. These instruments are built to see through the seawater and onto the sea floor. What we can get out of that are diagnostics over the entire 200 kilometers of West Hawaii that tell us how the coral is doing. And we can zoom in and see where the coral is alive, where it has unfortunately died, and everything in between along this coastline. And from those diagnostics, we can start to look at treatments. The treatments that you've heard about tonight are the treatments that are in play here in West Hawaii. We are particular fans of something called larval production, which we heard about, also known as sexual reproduction of corals. And we have built a massive nursery in Kailua Kona to do that. You're seeing pictures of this nursery in action, even as recent as last week during a major coral spawning event that took place here. How are we doing that? A cartoon looks like this. We're in the water, we're in the air, we're looking to diagnose the regions that are doing well, the regions that are doing poorly. Sometimes the corals don't need to come out of the ocean, many times. They just need a little bit of help in the ocean. I'm going to skip that. That's called coral reattachment. The high-tech stuff is bringing them into our facility. You heard about thermal resilience testing, you heard about sexual reproduction, and you've heard about larval production. One of our innovations is injecting these larvae back into the reef using vessels and aircraft and net systems that allow us to place them in certain parts of the reef. Where do you place them on the reef? One of our specializations is understanding how those larvae flow in the current along the coastline. Those arrows that you see in the image is that current mapped out really for the first time in 2025 in the kind of detail that'll let us pick the sites along the coast, put the larvae in, inject them in, and they will settle down current in the injection site and down current as well. It's a reminder, though, that all of this is about the people and the place, and the science is really the toolbox behind the community. Thanks a lot, and if you want to see what we're doing, check out akoakoa.org. Thank you.
Thank you, Greg. So important to see all those complementary approaches coming together. I'm wondering, how do you measure the success of some of those interventions, like putting larvae into the ocean? How do you know how that's working?
We use a variety of methods. You heard of one, the diver-based imaging. We do that over time, repeats transects to see corals coming back or not coming back and understanding that. And we're also experimenting with some natural dyes in the corals. They're temporary, and the corals will actually light up under a certain light. We apply a UV light to them on the seafloor and see that they're there. They've settled, as you heard about settlement, and that they're taking back that seafloor that we want them to take back. So there's a lot of methods, but those are two really good high-tech examples that are in play today.
Amazing. So with that dye, you can see which of the corals that settled came from your releases.
That's right.
Is that right?
That's one of the techniques. And they're very early part of their life cycle. They're very early part.
Okay.
Later, we use more standard imaging techniques, return to the reef, do the high resolution 3D imaging on the reef, and look at those reefs change over time.
Great. Well, thank you, Greg. We're going to bring all of our speakers back to join you here on the Lightning Talk stage. And before we turn to audience questions, I wanted to ask if any of you have burning questions for one of your fellow panelists to kick us off.
I have a question. Go ahead.
Great.
Yeah, I had a question for Trinity, I guess, because I know there are also heat tolerant strains of the Symbiodinium algae that live in the coral. So I was wondering if all the corals in your study had like the same strains of dinoflagellates, or how do you think the contributors of the genetic thermal tolerance from the coral itself versus from the algae, how do those like interact?
Yeah, so that's a great question. And, corals have, their big holobiont, and it's a very complicated system of lots of different players, driving their thermal tolerance. So specifically with the data I showed, that was with a species from the Caribbean, Acropora cervicornis, and they only host one strain of algae. And so while we do have that, like the strain and the genotype of the algae as a cofactor, there's actually not as much variation in the role of the algal symbiont with that species. But with another species we see in the Pacific, they host a lot more different genera, and we see a lot more variation driven by those genera. And so piecing apart the role of the host and the symbiont in how this is shared across many different species is sort of part of our work.
Very cool.
Amy, you had a question.
Yeah, my question's for Greg. It sounds like you work with a lot of really high tech gear and technology. So do you have to have a background in chemistry, biology, engineering? Like, what's the background of someone that does the work that you do?
The background nowadays is bringing people together from many different disciplines. I think that's probably my strongest current trait. But my core background was in evolutionary biology and in physics. So kind of having those fundamentals somewhere under the hood is helpful to me. But nowadays in this sort of program, I'm really looking at folks like you, looking at people who know how to do this, really specialize in high-tech contributions to your picture.
Right, great start. So there are a couple of audience questions related to spawning that I think a few of you could help answer. So Nadej, I'm going to start with you because our audience was intrigued that you mentioned that the spawning events are linked to the lunar cycle. Can you talk about that a little bit more?
Yeah, so the really amazing thing and also challenging thing about studying coral spawning is that you have so many species and they exhibit so many different like reproductive modes and strategies and timing, and so it can be hard to study those. So yeah, but for a lot of corals, they do spawn on certain days of the month. So like during the full moon or right after, or some of them do the new moon. And usually it's only a couple of specific months of the year. So maybe in the fall or in the summer. And those are usually linked, we think, to kind of the tidal heights and also the amount of daylight and moonlight that is illuminating the coral. So that's why we think that they are sort of sensitive to the lunar cycle, is because it has those impacts on the tides and on the amount of light. But there are really a lot of different ways that they do it. There are also some species that don't release their gametes into the water column and brood their larvae internally. So the a larvae come out in a more advanced developmental stage, and that can affect like how long they can survive in the water column and how far they disperse. So it's, yeah, coral spawning is fascinating, but it's also very, very complex and diverse.
And Greg, how do you manage that in your nursery? All of those complicated factors.
I have great engineers that manage it. I don't personally.
Is it tricky to get it right?
Yeah, I see it, I hear it. They are constantly tuning, retuning, reinventing themselves and their efforts and the equipment. This entire field is on a rapid nonlinear rise, and all of the topics we're talking about today are evolving so fast, including all of what we're talking about in your question. So without a doubt.
That's exciting. Hopefully we'll all learn a lot more quickly. To kick off the next portion, there was a question for you, Amy, related to, you mentioned that the research method you use is unique. Can you talk a little bit more about how that differs, or any more background on that particular method?
Yeah, absolutely. There are a number of different ways that researchers can collect data from coral reefs. I talked about mine, Greg talked about his. It all kind of depends on your resources and how many people you have. A common way that folks do coral reef surveys is still diving, but you have a slate, which is like underwater paper essentially, and a pencil. And so a lot of folks will manually write down what fish they see, what corals. What we use is a video camera. And the way that our video is unique is that originally in 2009, we started this project because we were interested in the fish. So typically when people do benthic or coral or bottom of the ocean studies, they're taking pictures and videos of the bottom, but our videos are facing out because originally we were looking at fish. The cool thing about saving all of these videos is that you can go back and ask other questions and extract data out of the videos that you have. So ours is a little bit different. It's unique in that it's a different way of facing it, but we have published on this so that others can also use this method as well. And that's another way that scientists like to share their methods, is to publish it and share so that others can repeat those methods as well.
That leads into another question we got, which was wondering if scientists share how they're doing this research and what they're learning, and learn from each other. And I already heard some of you ask each other questions that makes it clear that yes, you do learn from each other and connect. Does anyone have any other examples of how you connect with other coral researchers, or what you've learned that you've applied?
I'll just jump in and, so go for it. Go for it.
I think for coral work specifically, sort of what Greg mentioned about like community and getting involved with stakeholders and practitioners who are sort of in place in areas that have reefs and have corals is really important. So in my work, I've been lucky to like partner with the Nature Conservancy and the Hawaii like Division of Aquatic Resources and the National Marine Sanctuary out in Maui, that like making sure that as a scientist, even if you're maybe coming from an institution that's not located where the reefs are and you're going out to do that work, that when you do it, you don't just sort of go to the place, do the work, and then fly away and not engage with the community around it, because it's the people who are living and diving on the reefs every day and who are using it for subsistence and that kind of thing that the work really matters to. So I do think that, and it sounds like I've heard, a lot of people on this call sort of have partners and engage with those stakeholders like in the reef community. So that's important, I think.
What about you, Greg?
I was just going to add that in my career, I've seen the science community evolve, and perhaps it's because coral reefs are so endangered that the world of competition dominating academia is finally given away to collaboration. And it took time. And if you have my sort of view over some decades, you see the evolution, and it's heartwarming. You see a lot of people coming together at the conferences, across networks, regional and global networks. So that gives me a lot of hope that there's this sharing of methodologies and sharing of perspectives that maybe is driven by the fact that coral reefs are in such dire straits and they need our help.
Okay. There are a few questions about how colorful the corals are. So for you, Emma, first, comments about the beautiful green coral that you shared. Can you talk a little bit about why that one is green?
Yeah, so corals get their color from the zooxanthellae that live within them, and the lighting does play a factor into it. So if you're under really, really blue lights, it could make that green coral pop. And sometimes you can see on the reef there'll be different colors at different depths. So it just depends on how much light they're getting. They can get a different color. In a way you can look at them without having the blue interfere with it. We have you can have orange sunglasses. You can look at the corals and get a truer color of it. But that one was specifically green under that blue light for sure.
All right. And Trinity, there was a question, seeing the bleaching indicates that there's a problem. There's a question whether anything else about the appearance, like the color or anything else, can tell us anything about the coral's health or what's going on.
Yeah, so bleaching obviously is going to be the big one, and corals can actually, experience bleaching if it's too cold. So it's mostly because of rising ocean temperatures, but bleaching itself can be in response to other stressors. But also coral disease is something that can change the colors of these corals, lesions that are, slightly off of the original color or are, black sort of bands around the coral. And so usually we want to see that sort of red orange that the algae is giving the coral, and then things that are massively departing from that are typically a sign of stress. Another example is like the fluorescence that Emma was talking about. Sometimes corals fluoresce more as well under stress, because it sort of acts as like a sunscreen to incoming UV radiation that is more stressful under higher temperatures. So coral color is a very good indicator of how they're doing.
All right. So speaking of stressors, there were several questions about stressors in addition to the warming ocean temperatures and the diseases. So people raising questions about things like microplastics, cruise ships, lionfish, predators, sunscreen. So I'm wondering if maybe a few of you can share a little bit about other stressors that you've observed or that you're concerned about. You want to hop in?
I will try one. I have one that surprises people. On the last slide of my talk, there's some signage about pharmaceuticals. In our high tourism spots in West Hawaii, we have systems that we put into the reef that capture chemicals over some period of time, call it a month. And we'll put them in before, say, Christmas break and until after the New Year. And so high tourism time, and the material inside these mechanisms will collect up the different molecules that flow in the water column and touch the coral's exposed to. And we have discovered that there's a lot of pharmaceutical load from the tourism industry, from swimmers. And it gets into some sticky business here talking about it, but we've detected birth control, we've NSAIDs like acetophenamine and ibuprofen, things that really disrupt coral physiology. And most people know about the sunscreen issues and how in Hawaii we banned certain chemicals in certain sunscreens in 2016. And that was both important and symbolic. But there are other chemicals that we're bringing into these reef systems, and people should be aware of that and help us by eliminating that source.
For folks that don't know, you can buy now sunscreens that are labeled reef safe. And so these are more natural sunscreens that don't have the chemicals that block UV rays that help corals live. And so even here I live in Seattle, Washington, I still go swimming in the lake and in Puget Sound sometimes when it's not super cold. You can still use reef safe sunscreen. And then also just thinking about any other products that you put on your body before you get into the ocean, that will come off and float off into the water. So just being a little bit more cognizant of what you're bringing into the marine environment.
Thank you. Nadesh?
I was just going to add, just from my sort of perspective as a somebody who thinks about sound in the ocean environment, which is maybe not necessarily something that a lot of people are familiar with as a pollutant, but noise pollution on coral reefs, but also in the ocean more generally, is a growing issue that I think has only recently kind of started being recognized for its ecological relevance. So the amount of noise that we add into the environment from shipping and even in protected areas that maybe are only frequented by tourist boats, the motors from their engines can still be very loud. And it's sort of like if you were if your house is living right next to a busy train station or right under an airport, noise can be a stressor for animals. I was talking in my talk about how the noise, the natural sounds of the reef are very important ecologically. And so it can be a really positive thing, but you can also have sound be a negative stressor in other ways. So it's not as acute of an impact or as warming, and it may not cause some of the same really drastic effects, but you can have stress that can lead to animals being confused or unable to communicate, or in really extreme cases, things like really high broadband or amplitude signals can actually cause damage to animal tissues. So it's something to think about in marine protected areas.
Thank you. Emma, do you have any other stressors you want to mention or any ideas for what people can do to help protect and restore coral?
I mean, in the aquarium industry, we do have a lot of reef safe products as well. So like the glue, the epoxy that I talked about, it is similar to that sunscreen where it doesn't have the chemicals that you might use on land. So it doesn't break down in the water as easily and introduce those chemicals to the corals growing within a closed environment.
Great. That was one of the questions from our audience. How is that glue safe for the coral? So thank you for answering that. Trinity, any other tips for people who want to help coral?
Yeah, I think just echoing what everyone else has said, but also emphasizing a lot of this, I don't think that talking about, climate change and ocean warming can feel very big and very stressful. But there's so much at the local scale, things like these chemicals we're talking about or nutrient runoff that can increase the stress from climate change that these corals experiencing. And so there's so much that can be done at maintaining water quality at the local level that actually really helps and can help these reefs, make it through really bad heat waves and things like that. And so just emphasizing how much water quality really matters under these sort of larger climate events.
Okay. Thank you all for all those action ideas for all of us who love coral. Before I ask my final question, I'll give you a preview of what it's going to be. Everyone is curious what your favorite coral is or a coral you want people to know about. So you can think about that for a second while I ask Emma one more question. There's a question about fragging coral and whether you can keep fragging the same coral parent and then the child coral, and then that one, can you keep going indefinitely?
That would be the goal, to keep fragging over and over again and have this one really healthy parent coral and then eventually make babies from that coral, and you can make parents from that coral, and just it can be an endless cycle. So we can keep that coral healthy, alive, happy in all these different places and share it with other facilities if needed. And that would be the dream, to keep it over and over again.
And then one last question for you. How do you ship that coral to other aquariums safely?
I think you saw on one of my slides, there was a box and I had some little baggies in that. Basically the coral gets placed in a baggie full of like half full, 3/4 full with water. And that rest of the bag will be filled with oxygen to keep the water nice and oxygenated while it travels. And then you put them in the box, make sure they're standing upright and they have no way to flip over. And then we can ship them for FedEx, UPS, and hopefully they land safely and they can have the parents can send their children off and be thriving everywhere else.
Love it. Okay. Let's talk about a kind of coral that you all would like our audience to know about. Can we start with Trinity?
Yes, I think one of my favorite corals, or coral that I think everyone should know about, is its scientific name is Acropora palmata. It's called the elkhorn coral. It's in the Caribbean. I'm partial to it. It's what I studied during my PhD, and they can form these massive stands that look like, you know, elkhorn, and they just create these like massive fields that are, I think, some of the most breathtaking. If you can find some pictures of some of the remaining fields, I think that would blow some people's minds.
Well, I'll do some Googling after this. Amy, you're up next.
I think as a underwater photographer and videographer, I like to kind of zoom down into the really little tiny things. So I think cup corals are one of my favorites. They're just so cute and tiny, and they look like little flowers. So for anyone who doesn't know what that is, please Google image it.
Do those come in all different colors, or are they a specific color?
The one that I've seen the most is orange. It's like a very beautiful orange.
Okay. Nadej, what is your favorite?
Well, it's hard to pick a favorite. I think that every coral is, special and has its own values. But one that I was really fascinated to learn about were the mushroom corals. So these are Indo-Pacific in the genus Fungia, I think, like fungus. And some of these corals actually, they're very different from a lot of these other species we talk about, in that some of them live as only one polyp. So they're not necessarily a colony. There can be one polyp that grows really, really big. And I made a whole point in my talk about how corals generally aren't mobile at all as adults. And that's actually not true for some mushroom corals. So they don't necessarily attach to the reef. They can kind of they stay unattached, and therefore sometimes they can also extend their tentacles and move around, which I think is crazy because it is so unusual among corals. So yeah, I think the mushrooms are really, really cool.
It's fascinating. Greg, what coral do you think we should all know about?
Nadej took mine, so I'll go with my second favorite, close second, which is the antler coral, to get back to the ungulate style names. Antler corals in Hawaii, it's Passalopora grandis. It's forms these sort of candelabra style, really big. I saw one the other day that was easily the size of a Volkswagen Beetle. They stick out on the reef at about 15 meters water depth. So you're cruising along on the reef and it's undulating with all the rest of the coral. And then these crazy big antler corals are sticking out into the water column, and they just, they attract your attention and they attract a lot of fish.
Amazing. I would love to see that. Okay, Emma, bring us home with your recommendation.
Okay, my favorite coral is Caudagoniophora. It's also known as a flower pot coral. We have them here at Seattle Aquarium. We have a whole tank dedicated to them, and the ones we have are this beautiful pink color, and they have these beautiful flowy, flowery arms, which turn out really cool. And if you want a sneak peek of them, they are recently on the Instagram page on the Heated Rivalry post. So they look really beautiful, and they look even prettier in person.
All right. I encourage everyone to go look up all those coral. They all sound amazing and beautiful. And we're going to wrap up our episode. So thank you all so much to our speakers. You were all fantastic. We learned a lot. There were so many questions in the chat and lots of applause. So thank you for joining us. I am going to ask, I want to announce that we have our podcast launching. So we have a brand new podcast called Lightning Strikes Twice. And Amy, one of our speakers tonight, is one of our hosts for that podcast. So the podcast is a place where all of our speakers who only had 5 minutes for Lightning Talks get to dive deeper with our hosts and answer more of your questions. So if your question wasn't answered tonight, you should tune back in for those upcoming episodes of Lightning Strikes Twice. You can find it wherever you get your podcasts or through our website. And if you enjoyed tonight's free science event, we hope you'll consider a donation to help keep these fun events accessible to everyone. Our next episode will be about bioluminescence. That is coming up on July 23rd. So please mark your calendars, and we look forward to seeing you all again then.